guys, welcome back to Ferris Tutorials. In today's episode, we'll be looking at food, nutrition, and health. Past paper questions for the topic food preservation. CSEC past paper questions for the topic food preservation. Now, let's get started by looking at how this content is outlined in your syllabus. Different content area that falls under this section are the methods used to preserve food, such as cold preservation, and we may refer to what? The refrigerator, the freezer. We speak of heat, where we may speak of sterilization, canning, bottling, pasteurization. There's also the use of chemicals, where we may speak about vinegar, salt, and sugar, removal of moisture, physical, and also irradiation, which refers to rays passing through these packages so that they are safe and their shelf life can be extended. All right, have a knowledge also of suitable foods for each method, right? Preservation of various foods at home, such as fruits and vegetables. How can we preserve those at home? So we may speak of pickling, we may also speak of dehydration, freezing, refrigeration, right? Awesome. Also suitable foods such as food from animals as well, such as meat, seafood, milk, eggs. How do we store those, guys? Right? What are the different ways in which we can preserve those things? All right, we should be able to know for fresh fish, we may use what? Freezing, as well as salting can be used too in terms of codfish, where the salt will pull the moisture from the fish by the process of osmosis, so we get dehydrated, so we have dried codfish, right? All right, so these are the areas that you're required to know. Now let us look at some past paper questions, and we're kicking things off with the year 2017. Now let's look at this question. List 3 methods that are used for preserving food, and that is 3 marks. Now, even though it asks for 3 methods, we have more than 3 outlined here, but let us look what they are. So first, there is heat preservation. An example of heat preservation may include pasteurization, sterilization, ultra heat treatment, and also canning and bottling, right? So that is one. Next, we could also have dehydration as a method used for preserving food. There's also cold preservation, example refrigerating or freezing. There's also the addition of chemical preservatives, example pickling, and also the removal of air through packaging or modify atmosphere packaging. And an example of this is vacuum packaging, right? So you're only required to list 3 here in this question, but it is always good for us to have options from which you are to choose and become more, and the ones that you're comfortable with, right? So we have heat preservation, example canning and bottling. There's also dehydration, 2, and 3 we could have cold preservation, example freezing. Good. And we have 2 additional examples there of methods of preservation that we could have chosen from as well. All right, moving along to 2016. Explain why each of the following procedures are important when freezing green beans. 8 marks. Now the procedures are washing, blanching, sealing the package, and also rate of freezing. Good. Let's read the question again. Explain why each of the following procedures are important when freezing green beans. Good. Now, why do you think it is important for us to wash before we even begin to blanch or to seal the package or to think about the rate of which we're freezing, right? So 1, if your response was for washing to remove dirt and any other foreign matter from the beans, you are correct. Now, as it relates to blanching, right, blanching preserves the color, the freshness, and flavor of the beans. Good. And it also prevents ice crystals from forming in the beans as well. Now guys, we're looking at why is it important for us to seal the package. So after the beans have been blanched, why is it important for us to put it in a Ziploc bag or a container in which all the air or most of the air has been removed, right? Now this is important because it prevents the bean from being freezer burn. Good. As exposure to air will cause the drying out of the beans and also leads to the destruction of the nutrients as well. Good. Now, rate of freezing is also very important, as very rapid freezing will prevent ice crystals from forming, which can damage and destruct the cell walls of the beans. Good. So if the rate of freezing the beans are low, then large ice crystals will form. So to prevent that, we have to ensure that the rate of the freezing is taken into consideration to prevent large ice crystals from forming, which will destruct the cell walls of the beans. All right. Let us look at 2015. John is responsible for the preparation of a variety of preserves for a stall at his school fair. State 2 reasons for preserving foods, right? And we have 3 reasons here for you. So 1, to prolong the shelf life of food. 2, to make foods available when they're out of season. And 3, to make food available to other territories, foods that are not found in their country. Good. Next, define the term sterilization. 2 marks. The term sterilization denotes the complete destruction of microorganisms by heat, right? So for example, during bottling and canning, whether it may be juice, fruit juices, or milk, or other beverages, or canned fruits or vegetables, right? Sterilization, whether it be in the jar, bottling, or canning, it is important for this process to take place so that microorganisms are destroyed and to provide a sterile environment for the food where microorganisms cannot reproduce. Now we're still on 2015. Explain the scientific principle involved in the prevention of the growth of microorganisms in each of the following. There is guava jam, pickled cucumbers, and salted codfish. All right. Now we're supposed to explain the scientific principles. All right. Now, as it relates to guava jam, which preserve is used? Right. Awesome. If your response was sugar, you are correct. All right. Now, along with sugar, there's also acid as well. Right. So we may say altering the pH because we use some form of acid. So based on the fruits that are used, they should be, for jam making, they should be high in acid, right? So for example, even if they're not high in acid, you may find out that the recipe will tell you to add some lemon juice or some lime juice just to kickstart the gelling process of these jams or jellies. All right, so we may use altering the pH because there is some acidic content that is present, and also the use of chemicals, right? Because sugar is considered as a household chemical as it relates to food preservation. All right, so we may say altering the pH as well as use of chemical. So sugar, acid, and pectin, right? So these 3 ingredients lower the pH of the preserve and bind available water, thus creating an environment in which the growth of microorganisms is retarded. Good. Now, as it relates to pickled cucumbers, Altering the pH is a scientific principle here. Now, the pH of the food will be lowered so that the food environment becomes too acidic for microorganisms to grow. And you know, the preserve that is used here is the vinegar, spice vinegar. When we're making pickles, right, the liquid that is used is spice vinegar, and it is acidic. Therefore, it creates an environment where microorganisms cannot grow. All right, the next one is salted codfish. And the scientific principle here is the removal of moisture. So the salt pulls the moisture from the codfish by the process of osmosis, which leaves the cod dehydrated. So the same thing that we call saltfish, guys, we're referring to here as the correct name as salted codfish. So just for you to be clear on that. All right, let us move on to 2014. Discuss the scientific principle that explains the removal of excess water from cucumber during pickling. Good. All right, so the spiced vinegar is more concentrated than the solution inside the cell of the cucumber. Therefore, the water will pass out of the cell through the process of osmosis. So it moves from what? A higher concentration to a lower concentration. Now, the movement of the water from a high to a low concentration through a semi-permeable membrane. And what a semi-permeable membrane means is that not all of the things will pass from the cucumber into the spice liquid, right? What it does is to remove the moisture, some of the moisture content from the cucumber, which enables the production or the growth of microorganisms. All right, so the nutrients will not be lost, but the water itself will be pulled away from the cucumber and in an environment in which microorganisms cannot reproduce or grow. Good. 2013, Petra received a surplus of green beans and is advised to preserve some by freezing for later use. Name 2 methods other than freezing by which she may preserve the green beans, right? And that is 2 marks. So freezing is out of the way, so we must come up with some other methods of preservation that can be used, and the ones we have here are canning, pickling, and also dehydration. Good. Name 1 preservative that can be used when preserving green beans at home. 1 mark. And the correct answer there is vinegar, right? So name 2 method, it could be pickling and dehydration. There I just added canning as a bonus, but you know, if she's at home, it is highly likely possible for her to carry out the procedure of canning, right? So let's say pickling and dehydration. So the 1 preservative that may use right there is vinegar. Good. Give 1 reason for the use of the preservatives named above. So it is asking us, why did we choose the vinegar? So the vinegar can be used to create pickled beans, which will alter the pH. So the scientific principle there is for these green beans, we're altering the pH, which means that the environment will be too acidic for microorganisms to enter, to grow and reproduce, right? Now, the pH of the food will be lowered so that the food environment becomes too acidic for microorganisms to grow. Now, continuing with another aspect of 2013, Petra is advised to blanch the green beans before freezing to inhibit enzyme action. Define the term enzyme action. Good. Now, this is the process by which enzymes act on food products by speeding up the process of decay by breaking down tissues and other components of the food. So what will take place? Browning may take place, oxidation, and also ripening. And all of these are the action of enzymes. Good. So enzyme action may be defined as the process by which enzymes act on food products by speeding up the processes of decay, just as how enzymes help us to break down food and also in digestion. It speeds up the process of decay in food, and what it does is to break down the tissues and other components. So as soon as the meat is slaughtered, as soon as the fruit or vegetables are picked from the tree, then the enzymes are activated. And if these are not controlled, if conditions are not controlled, then the enzymes will continue to break down the food until it deteriorates, right, or decay. Next question, identify 2 undesirable effects of enzyme action on green beans if they are not blanched before freezing. So it asks for 2, but we have 3 here. So discoloration due to inactivation of the enzyme. So if the green beans are not blanched before freezing, then what? Enzymes are not deactivated, and therefore it will continue to work to break down the tissues of the green beans, which can lead to decay, right? Or you see that discoloration, it starts to look brown, right? Now, microbial activity in the beans can also lead to textural change. So on thawing, the beans become what? Soft, and it may be soggy. Good. Loss of nutritional value due to inactivation of enzymes. So remember, the process, the function of enzyme is to break down. So if they are not inactivated, what will happen? Discoloration of the beans will take place, and also the nutritional value of the beans the nutrients will be destructed, or they will be lost. All right. Now moving on, again, identify 2 procedures to be followed before placing the blanched green beans in the freezer. 2 marks. So after they have been blanched, what should we do? So 2 procedures include 1, drain well, as frost or ice crystals which may appear on the surface can affect the texture and appearance of the green beans. So after they have been blanched, shock, and we drain them well before we store, right? Now the next procedure is to seal the package to prevent loss of moisture from the beans, or to reduce the amount of air that comes in contact with the beans, as these will cause also destruction of the nutrients as well as ice crystals being formed. Give 1 reason why each procedures identify above. Good. So the reason we're given here: when normal green beans are frozen slowly, large ice crystals form on the cell and ruptures the cell. When they are thawed, the ice crystals melt and the beans become soggy. So this is the reason why it is important for us to drain the beans before they are stored, and also to seal the packaging, right, before we freeze them as well. Skipping on over to 2009, as part of your SBA project, you are required to conduct research on food preservation and share your findings with your class. Define each of the following terms used in food preservation. So there is bottling and also blanching. Now let us look what these definitions are. Bottling may be defined as the process of preserving foods, in particular fruits, vegetables, and meats, by packing them into glass jars and then heating the jars to create a vacuum seal that kills the microorganisms that would create spoilage. Good. Let's go again. Bottling may be defined as the process of preserving foods, in particular fruits, vegetables, and meats, by packing them into glass jars and then heating the jars. So when we speak of heating, we may speak of sterilization or pasteurization, to create a vacuum seal that kills the microorganisms that would create spoilage. Good. Our next term, blanching. Blanching may be defined as the cooking process where food, usually a vegetable, is scalded in boiling water, removed after a brief time interval ranging from 1, 2 to 3 minutes depending on the type of vegetable, and finally plunge into ice water or place under cold running water, and this process is known as shocking. And what this does is to halt the cooking process to prevent the overcooking of the vegetables and also to prevent nutrients from being lost. All right, now at your presentation, a classmate noted that after thawing pigeon peas, which are gungo beans or gungo peas, they became soft. Explain what may have caused this to occur. 2 marks. Now, why do you think after freezing the pigeon peas or the gungo peas, they became soft? Can you tell? I know you can tell. So the pigeon peas was not blanched before freezing, right? And therefore, ice crystals formed inside the cell walls and caused it to expand and may even ruptured, right? Ruptured. As a result, the texture is damaged, thus causing the pigeon peas to become soft after thawing. Now, think about it. Have you ever placed any vegetable inside your refrigerator at home because you have a surplus, because you want to preserve it, but you didn't blanch it? What do you notice after thawing? Now, after thawing, they become soft and they become soggy because ice crystals form within the cells and they expand until they burst. Good. And therefore, when these ice crystals have been melt, then you notice that the what we call puny. You notice that the vegetables are soft and soggy, and in most cases the enzyme action take place too because they weren't blanched. All right, so you may notice that they look a little brown. All right, now we're looking at jam making. This question was taken from 2016. There's not so much emphasis on jam making anymore since the syllabus have been changed, but persons have been asking for jam making questions, so I've included a question here for you to have an understanding of what jam making is, because even though there's not an emphasis on jam making, you may just get a question that asks you how is it that you can preserve a surplus of mangoes or a surplus of strawberries or apples, whatever the case may be. All right, and one such method of preservation may be what? Jam making, right? Where we use the sugar, acid and also pectin. All right, let's go to the question.
Vanessa is making a batch of mango jam. List 2 tests that Vanessa could use to determine when the jam is set. 2 marks. So we have 3 methods here listed for you, 3 methods of testing if a jam is set, and the first one is the flake test. There's also the saucer test, and also using a thermometer to test the jam for an ideal temperature of 105 degrees Celsius or 220 degrees Fahrenheit, right? Now the next part of the question reads, describe the procedure for any one of the tests listed above. 2 marks. So we're starting off with the flake test. So how this is done? Now, as the jam thickens, a wooden spoon is used to scoop a small amount of jam out of the pan. The spoon is then lifted above the pan about 12 inches and tilted horizontally. And after we have done this, now we're watching to see how the jam runs back into the pan, right? Now what we're aiming for is a cascading sheet-like fall rather than a single line of runny jam. Good. There's also the saucer test. And you can also let a spoonful of jam sit on a cold plate for 30 seconds. Or we could put the jam directly on the cold plate for 30 seconds and then we push back with our finger or a spoon. Now, if the mixture wrinkles up, then we have reached the setting point for the jam. All right, and the final test here is a thermometer. Now we can use a candy thermometer to test the temperature of the jam. Take the temperature when the jam has reached a rolling foamy boil. Now you are required, what you're looking for now is a temperature of what? Awesome, 220 degrees Fahrenheit or 105 degrees Celsius. Now at this temperature, the jam will set nicely.
Final set of questions. List 2 guidelines other than pectin content that Vanessa should consider when selecting the mangoes. So 1, the fruit should be just ripe, so it shouldn't be overripe. It should be just ripe so that it has a good amount of pectin and acid present. Good. Ensure the mangoes are of good quality, free of signs of deterioration. Next question. The mangoes Vanessa selected do not have sufficient pectin. Suggest 2 ways in which the pectin content could be enhanced during the preparation of the mango jam to ensure that the mixture is well set. So what she could do is to add commercial pectin, or she could add some lemon juice or lime juice, which is acid, to help the setting process, to kickstart the setting process of the jam. You may also find that foods that are rich in acid are also high in pectin. Good. And that's another reason as well for using the lime juice or the lemon. You are awesome. You've made it to the end of the session. Please remember to subscribe, turn on your post notification bells so you can be notified when there's another upload. Watch those ads, leave a like, a comment, and most importantly, share with persons who you know will